The 8 steps of heme synthesis. Heme synthesis is an 8-step biochemical pathway that produces heme, an essential iron-containing molecule primarily used in hemoglobin. This highly coordinated pathway takes place sequentially between the mitochondrial matrix and the cytosol of human cells. The primary goal of the pathway is to construct a porphyrin ring structure that is capable of chelating a ferrous iron atom. The resulting heme molecule is vital for oxygen transport, cellular respiration, and drug metabolism. What is the rate-limiting step of this pathway? The rate-limiting step of heme synthesis is the very first step. The condensation of Succinyl-CoA and glycine to form delta-aminolevulinic acid. Molecular mechanics of this reaction is: it occurs in the mitochondrial matrix, conducted by the enzyme aminolevulinic acid synthase, in the presence of cofactor, pyridoxal phosphate, also known as vitamin B6. How does vitamin B6 deficiency impact this specific step? Vitamin B6, specifically its active form, pyridoxal phosphate, is the essential cofactor for aminolevulinic acid synthase, the rate-limiting enzyme of heme synthesis. Without B6, the very first step of the pathway cannot occur. So, what are the clinical consequences of B6 deficiency? When vitamin B6 is deficient, heme production drops drastically, leading to specific medical condition known as sideroblastic anemia. In this disease, iron enters the developing red blood cells normally, but because there is no protoporphyrin heme to bind to, the iron traps inside the mitochondria. This creates a distinctive ring of iron around the nucleus, visible on a blood smear as ringed sideroblasts. Let's discuss the pathway steps and locations of heme synthesis. The complete, step-by-step chemical transformations of heme synthesis occur across 8 distinct enzymatic reactions. Step 1. Formation of delta-aminolevulinic acid. Location of this step is mitochondria. In this reaction, Succinyl-CoA and glycine condense together by enzyme aminolevulinic acid synthase. As already discussed, this is the rate-limiting step and requires vitamin B6. Step 2. Synthesis of porphobilinogen. This reaction occurs in cytosol. In this reaction, 2 molecules of aminolevulinic acid condense and dehydrate to form a single pyrrole ring, in the presence of enzyme aminolevulinic acid dehydratase, which is also called porphobilinogen synthase. This enzyme is highly sensitive to zinc displacement by lead poisoning. Step 3. Synthesis of hydroxymethylbilane. This step also occurs in cytosol. In this reaction, 4 molecules of porphobilinogen link together in a linear chain by enzyme hydroxymethylbilane synthase, also called porphobilinogen deaminase. Step 4. Cyclization to uroporphyrinogen 3. This step also occurs in cytosol. In this reaction, the linear hydroxymethylbilane chain is closed into an asymmetric, pyrrole ring structure by the enzyme uroporphyrinogen 3 synthase. Step 5. Decarboxylation to coproporphyrinogen 3. This step also occurs in cytosol. In this reaction, the 4 acetate side chains on uroporphyrinogen 3 are decarboxylated into methyl groups, in the presence of enzyme uroporphyrinogen decarboxylase. Step 6. Oxidation to protoporphyrinogen 9. Now this step also occurs in mitochondria. In this reaction, the coproporphyrinogen 3 enters the mitochondria, where 2 propionate side chains oxidize into vinyl groups by enzyme coproporphyrinogen oxidase. Step 7. Oxidation to protoporphyrin 9. Location is same, mitochondria. Reaction involves the central ring structure of protoporphyrinogen 9 loses 6 hydrogen atoms to become a conjugated, light-absorbing porphyrin ring, in the presence of enzyme protoporphyrinogen oxidase. Step 8. Chelating iron to form heme. Its location is also mitochondria. In this reaction, ferrous iron is inserted directly into the center of protoporphyrin 9 by the enzyme Ferrochelatase. Clinical relevance. Tight regulation of this pathway is crucial. Genetic or acquired defects in any of the 8 enzymes result in dangerous accumulations of toxic metabolic intermediates. These conditions manifest clinically as porphyrias, causing neurovisceral or cutaneous symptoms, sideroblastic anemias, or lead poisoning.